I'm Sarah Donoghue. I come from Melbourne, Australia. So I've had a big trip over here to come and speak to you. Um, I am a PhD student supervised by Anna Voss and David Amor. Um, and I do my work out of the Murdoch Children's Research Institute. So just with my disclosures and conflicts of interest, I've just got government funding for my PhD research. I don't have any particular associations with pharmaceutical companies and no conflicts of interest. So I guess people know a lot about KAT6A in terms of many of the different functions that it performs. We know that it forms part of a multi subunit complex of proteins which are responsible for putting acetyl groups on histones, and these histones are strongly associated with neurodevelopment. It's got an important role in transcription, and it also is a regulator of P53, which is important for cellular mechanisms, particularly around cell program death, and it also binds to transcription factors RUNX1 and RUNX2. We also know that from recent work that you can get rescue of loss of function variants in KAT6A mice models by overexpression of KAT6B, which is work that Maria Bergamasco had recently done out of Anna Voss's lab. So I guess when evaluating how this condition impacts on neurodevelopment, there are many different ways that we can look at that. And we're quite lucky that we've had a lot of researchers today who are looking at that in many different ways. So with neuropsychological testing and speech and language testing, there's been some reports on neuroimaging abnormalities in this condition, and also studies on animal models, and this is the area of interest that I have at the moment, looking at mice models. In my lab as well, we're generating human cortical neuron models, and for the work that we're currently doing is looking for a biomarker. So what do we know about the neuropsychological profile studies in KAT6A syndrome? So Mia St. John, who's previously come to this conference, had done a PhD looking at speech and language in patients with KAT6A, and we knew that children, and this probably captures the more severe end of KAT6A, were often minimally verbal, and in those who were verbal, often they would have complex speech issues with apraxia and phonological disorders, and in that particular cohort, intellectual disability was common, and a third of them would have a diagnosis of autism. There's been more recently published information looking at the cognitive profile, and I know that studies are ongoing for this particular team, and they found similar things: impaired non-verbal communication, similar to receptive language, high rates of inflexible behaviours, and difficulties with global adaptive deficits. I guess this sort of captures probably the more severe end, but I know that we're all meeting families who have children who are more mildly affected. With the neuroimaging studies in this condition, so with MRI, it can be completely normal. You can have thinning of the white matter and corpus callosum, so non-specific changes, and you can also get cysts that appear and then disappear. And some people have had pituitary abnormalities reported. So once again, nothing specific that we can kind of monitor to see if there's change over time. With the studies on the mouse models, this is work that Anna Voss had done early in 2009, where she'd generated models where they had a loss of function. So this is where she'd knocked out the coding parts of the gene in exon 3 and 4, and found that there were abnormalities in the expression of Hox gene loci. So those were the genes that were influencing different segment development, particularly in the ribs. And you can see in the picture that I've put up that the KAT6A mouse The pup in the picture B, and you can see that that mouse is a lot smaller than its unaffected wild type beside it, and it's also got some differences in the way that the spine is formed. What they'd found is that there was some abnormalities in how histones were modified. So in particular, there was low acetylation, so the acetyl groups weren't being put on as much of H3K9, and correspondingly there was increased methylation of H3K9. So that seemed to be quite important for development, and certainly we've seen in other conditions you can get abnormalities in acetylation and methylation for neurodevelopment. She'd actually treated the mice with retinoic acid, which reversed the segmentation abnormalities, which is interesting, but I guess it's not necessarily a practical treatment in humans. There's been some recently published work just last year out of Baltimore, where they had been studying the behavioural phenotype of a KAT6A mouse. There had also been some work in the Voss lab, but they hadn't yet published that. But I think they'd found very similar things with the behavioural tests that the mice had done. So there was normal motor activity, but what the mice had exhibited when they put them through some of their behavioural tests was less anxiety. There were some deficits in recognition memory, and they also had some issues with spatial learning. So I'm not really a mouse lab scientist, but I know that often they will do things like water mazes where they try and locate a maze, and that's where they didn't locate it as well. That's how they'd worked out that the spatial learning was impaired. You can also see, similar to the mice that I put up before, the mature mouse in C, you can see that the KAT6A mouse beside the wild type is a lot smaller than with the growth. This mouse is comparable to the Voss mouse because they also knock out similar exons. So they've done some further analysis of the hippocampus of these mice because it had a look, and it looked like that the neurons in this area seemed to be quite important. There was normal morphology, so grossly when you looked at the brain, the hippocampus looked normal and then a number of cells looked normal, but the mice had similar memory deficits when they generated a special model. What you can see in the picture is there's a reduction in something called spines. So with the neuron, it has a body, and then it has a big tail called an axon, which conducts the impulses. And on that tail, they have specialized outpoutings called spines. And with those spines, they're very important because they contain a lot of metabolic material that's important for transmitting information. And what we often see in intellectual disability models is a reduction in spines, and that's what they'd found in these models. So it was quite a subtle difference. And when they'd done more studies looking at gene expression, they found one gene, RSPO2, seemed to be quite important, and it regulates, KAT6A regulates the transcription of this particular gene by modulating acetylation. So I guess this is where they thought the hippocampus is involved in memory. Perhaps this gene might be particularly important for some of its interactions within cells. And this is what we're interested in when we're looking at the neuron, to see what is it about neurons that makes things different with messages, and how does that influence the neurodevelopment in KAT6A. So with this particular gene, which codes for respondin 2, it's expressed and seems to be quite important in neurogenesis, so when we're making new neurons, and also for the differentiation of neurons as well. It's also got a really important function for regulating something called a Wnt pathway, WNT, and what it is supposed to do is enable a protein called beta-catenin to help with regulating gene expression. So what we're starting to realise is it's not just particular lipids or fats that are or amino acids that are causing difficulties. There's also some complex protein interactions within the cell, and we're trying to tease these out a little bit more to see if there are measurable things in blood where we might be able to use them for biomarkers. So what I've been doing in my PhD is we were looking at neural stem cells. So these are very early cells. So we had 3 mouse lines. And the reason we were studying embryonal models is because we really wanted to see if those changes were accentuated when we had a loss of function on both alleles, so a homozygous knockout, and a heterozygous model and a wild type. So we were hoping that changes might be more accentuated in that homozygous model. And so we collected stem cells, we looked at gene expression, we looked at metabolomics, and we also collected plasma from aged mice as well, up to 6 months of age, because that's the age where they might start to show some differences in their behaviour. And we've done metabolomics on that and neurofilament light chain, which is a protein that is expressed when cells die. And we're also looking at proteomics. I don't have the full results for the plasma studies yet. We're still looking at those, but I do have a little bit of information about the neural stem cells. And as part of the study that was going on for the behavioural phenotypes that Anne was doing, we've also got mice treated with carnitine, where we're hoping to analyse the brain once we find out what particular proteins are dysregulated. So for the neural stem cells, when we looked at the RNA expression, we didn't find similar things with respondin. But there were networks of genes that appeared to be dysregulated, and these seem to be in regards to DNA expression. There's also a group of genes looking at the, it's called a phosphatidylinositol pathway. There were a few genes that were dysregulated in that. That's a really important pathway within the neuron that does communicate with the neuron about cell growth, cell fate, so whether cells die or not, how big they grow, and it's a really important pathway that's now being increasingly implicated in intellectual disability. With metabolomics, we found one lipid that was abnormal, so we're hoping to prove that in plasma. And that was statistically significant. I guess it's a little tricky because it's hard to get something that's statistically significant, and maybe sometimes it's better looking at the network of what's happening. So on my next slide, I've got the top 36 compounds in my wild type versus my total loss of function model. So you can see that in red, there was only one that was statistically significant. But if we're looking at the Krebs cycle, you get the impression the orange is the compounds that are reduced, but perhaps some of those compounds leading into the Krebs cycle are reduced, and maybe the cell is compensating and driving towards acetylation. What we're also looking at doing is generating human cortical neurons and looking at those at different time points to do metabolomics, lipidomics, and proteomics. And here's some information that Jordan Wright, who's one of my collaborators, has been working on. He's been characterising what do the neurons look like under the microscope. And what you can see on the screen is where he has looked at the neurites, so the outpouchings from the neurons, and this is where he's removed the cell bodies, and he's taken pictures where you can see that there are reduced neurite lengths in KAT6A untreated, but some of those can be rescued with some treatments. So he's looking at many different compounds, and there's reduced synapses also, so you can see that in the bottom figure. and that there is some reduced acetylation, around about 50% in KAT6A models. So these are the cortical neurons that we're hoping to do a few more studies on, to have a look at what the gene expression and also some of these metabolomics looks like, and proteomics, to see if we've got any clues into what causes the pathology where people get differences in their neurodevelopment. So I guess that's our journey for searching with the biomarkers. There's a space then where neurons operate in, where they're maintaining what they need to keep functioning properly. And then there are situations where they get stressed, where things are dysregulated. So we're hoping to find chemicals that are produced in that space. We already know from some of our research already, at least in the mice, that they don't have any markers of cellular death when they're aged. So we're really looking in that chronic stress space to see if there are any clues for biomarkers. And the extra, I guess, diagram I've got is that we know from cancer studies where there are abnormal pathways expressed, and I'm particularly interested in the INK4 A pathway and the PI3K AKT pathway. So that's the phosphatidyl inositols that I was talking about before, because we know that those pathways, when they're dysregulated, can impair neurogenesis and problems with synapsing. And we know that from studies in cancer where people have had a loss of function of KAT6A, these pathways are dysregulated. And we think that this is really important for the development of neurons. I just wanted to acknowledge the rest of my lab. So Professor David Amel, who's one of my supervisors, and Paul Lockhart, Jordan Wright, who helps me, and also Professor Anna Voss, who has retired not that long ago, and her research team, Samantha Eccles, who is quite helpful with helping me with these mouse samples, and also our biostatistician, Brendan Ansell, who's been helping me as well.
So
Thank you.
Okay.
RNA transcription result, and you mentioned that there were no significant. What is it for a whole genome called transcriptomics or tissue?
So that was just purely off the neural stem cells that we'd isolated, that we got the whole transcriptomics for.
Yeah, we'll work in finding always 600, which like it's a found blood. And when we overexpress in the wind, that is a wind use that. When I look at your paper, because the axon 2 is also down, it throws me off, which means axon is part of the destruction of the complex. So I'm still not totally clear in the deficiency model, is wind really down or and it may be an oversimplification, but what can you just expand on your thoughts on what you think?
Yeah, so I'm hoping that we can actually look at networks of proteins expressed, which will help us answer this question a little bit more, I think, and that's the same with the genes. So what we're hoping to do is look at the overlay of the mRNA expression and also the proteins expressed to work out whether we do have something, a destructive process happening or not.
We isolated the neural stems, but then also you have it there with the whole, so depending on differentiating them into neurons, trying to take them for the mice.
So with the mice, we've just got the neural stem cells. That's all we've isolated for that particular project. But for the cortical neurons, we're going to look at, I think, day 4. and then a couple of different time points to when they should be mature. So we're looking at the mRNA for those different time points and the metabolomics and looking at proteomics as well. So
It's like most, I just
yeah,
stem cell, stem cell within the general cell in the fish, and show that they're just no matter what.
yeah, yep. So with the neural stem cells, they're isolated into, so they're, my understanding is that they are progenitor cells, and they were isolated into a little ball for all of the different samples. For I think it was 10 to the power of 8 is what we were collecting. Yeah.
Great.
Thank you.